So we can add in our understanding of how translation works by adding some sequence components to the mRNA molecule. The process of translation is carried out by the ribosome. And this is a process that takes the code from the open reading frame and converts it into an amino acid sequence or a protein. And each of these little circles is representing an amino acid in the protein sequence. Now remember that the open reading frame is defined by a start codon and by a stop codon, and I've rewritten these codons with U in place of T because it's mRNA. The start codon actually codes for an amino acid, and it is always methionine. And then each amino acid that follows corresponds to a certain codon in the open reading frame sequence. The stop codon does not code for an amino acid and simply signals to the ribosome to disassemble and stop translation. So how does the ribosome know to start translation at this particular AUG? So this is 3 of the 4 nucleotides that are found in nucleic acids. You can imagine that there are many AUG sequences across this mRNA molecule. Well, similar to the promoter, there is a sequence upstream from the start codon that the ribosome will recognize and bind to. And it is located 4 to 9 nucleotides upstream from the start. And the consensus sequence is AGG, AGG. This is called the Shine-Dalgarno sequence. There's a couple of other aspects of the mRNA that I wanted to point out to you. First is that there's sequence that is part of the translated region, and that corresponds to the open reading frame. And then there are untranslated regions. And so this is not translated and is referred to the 3' UTR, or untranslated region. And the other untranslated region contains the Shine-Dalgarno sequence. We call that the 5' UTR, or we call it the leader sequence. And this has important signals that, again, are recognized by the ribosome so that it can initiate translation at the start codon. Okay. So we can imagine, we move some of these annotations, we can add in the ribosome binding to the Shine-Dalgarno sequence. I'm just going to remove those for just a minute. The first part of the ribosome that will arrive is the 30th subunit. Of the ribosome. And ribosomes are mostly made up of ribosomal RNA, including the 16S ribosomal RNA. And it has a sequence that is complementary to the Shine-Dalgarno sequence. And so the Shine-Dalgarno sequence will base pair with each of these nucleotides of the 16S. Make the 16 knots maybe just a little bit bigger. And it positions the start codon in the P site of the 50S ribosome, which is where the first tRNA will arrive and bind to the start codon and begin translation.